Hello, I am Dr. Marathe from Department of Amphibolism. Today, I will try to explain the nature of TCA cycle, especially TCA cycle. So TCA cycle, when we try to study the TCA cycle, generally it is the oxidative nature of TCA cycle, means catabolic nature of TCA cycle. Catabolic nature of TCA cycle. Okay. Means whatever the end product of the glycolysis, that is pyruvate, through a pyruvate dehydrogenase complex, it is converted to acetyl-CoA. Acetyl-CoA enters into the TCA cycle along with oxaloacetate, okay, and various intermediates are formed through 8 reactions. So there are 8 reactions in the TCA cycle, and the 2 carbon which are channeled through the TCA cycle, they are evolved in the form of carbon dioxide in 2 steps, okay, and whatever the electrons are removed, those electrons, they are accepted by NAD or FAD, it's reduced to NADH plus H plus or FADH2, donated to electron transport chain. When they are channeled through electron transport chain by the process of chemiosmotic mechanism, the energy is released, okay, and that energy is trapped in the form of ATP, okay. That is one purpose. Means when we talk about the TCA cycle with respect to oxidative or catabolic nature, the 2 purposes are there. The carbon dioxides are evolved, and second is what? The energy is generated. But I am going to discuss today the another nature of TCA cycle. That in another nature is called as anabolic nature. Anabolic nature. Anabolism. When I say anabolism, it is nothing but what? It's a construction of complex molecules from the simpler one. Means in short, this is nothing but what? The synthesis process. When we talk about the catabolism is nothing but what? It's a destructive process, it's an oxidative process where the energy is released, okay, and electrons are removed. In case of anabolic nature, electrons are accepted, energy is used, okay. So we will try to learn the another nature, dual nature of this TCA cycle, and that is the anabolic nature. So when we talk about the TCA cycle, in the TCA cycle, what happens generally? Acetyl-CoA, CH3COO, SCOA, along with oxaloacetate, COOH, COO, CH2, COOH. This is oxaloacetate. Through the process of condensation, what happens? The enzyme citrate synthase, in which COASH is removed and it forms isocitrate. It forms a citrate. So citrate structure is what? COOH, CH2, COOH, again CH2, COOH. Okay. Now this is isocitrate. Do you know this isocitrate? It can be a precursor molecule for the synthesis of fatty acids and sterols. So from the isocitrate, fatty acids and sterols are synthesized. So that is what the anabolism means. One of the intermediate of the TCA cycle, that is citrate, it acts as a precursor molecule for the synthesis of various fatty acid chains and for the synthesis of sterols. This is what anabolic nature. Okay. Then another intermediate of TCA cycle is what alpha ketoglutarate. Alpha ketoglutarate, alpha KGA. Alpha ketoglutarate, and I will draw the structure of this alpha ketoglutarate. That is COOH, CH2, okay, CH2, C double bond O, COOH. This is alpha KGA. Now, do you know this alpha KGA, alpha ketoglutarate, it can be used for the synthesis of one of the amino acid. It is glutamate. Glutamate, which is the amino acid. Means this is the intermediate of TCA cycle, which has oxidative nature, means catabolic nature. But this intermediate can be used for the synthesis, or it can be used as a precursor molecule for the synthesis of glutamate. Okay, and from the glutamate to the glutamate family, other amino acids can be synthesized. These are glutamate, proline, glutamine, proline. Okay, and this glutamate, it can be used for the synthesis of the one of the most important intermediate of nucleotides, purines. So two kinds of nitrogen bases are there, purines and pyrimidines. So purines are synthesized from the glutamate. Okay, and glutamate is synthesized from what? This alpha KGA. So this alpha KGA is a precursor molecule for the synthesis of glutamate, which is amino acid. And that from the glutamate, various amino acids are synthesized, as well as it is also involved in the synthesis of purines. Another intermediate is called as succinyl-CoA. Succinyl-CoA. SUCCINYL. Succinyl-CoA. The structure is COOH, CH2, C double bond O, high energy bond SCOA. Now, do you know this intermediate succinyl-CoA? It is involved in the synthesis of, that can be used, or it can be used for the synthesis of porphyrin rings. Porphyrin. All of you know that this porphyrin ring contains a tetrapyrrole structure. Okay, so these porphyrin rings, these are the part of heme. These are the part of heme, HEME, as well as in the chlorophyll molecule. These porphyrin rings are present. What? Chlorophyll. So, when this chlorophyll molecule or the hemoglobin is synthesized, they require a porphyrin ring. That porphyrin ring is actually synthesized from the one of the intermediate of TCA cycle. It is succinyl-CoA. So this is the amphibolic nature. Okay. From this, another intermediate of the TCA cycle which can be used, that is what? Malate is there, and oxaloacetate. From the oxaloacetate, PEP can be formed. PEP, phosphonol pyruvate. Okay, by the enzyme kinase. Okay, by the enzyme kinase, from the oxaloacetate, phosphonol pyruvate is formed. This phosphonol pyruvate, through the reversal of glycolysis with certain bypass mechanism, it is converted to glucose. This glucose can be synthesized from the intermediate, that is oxaloacetate. So, in other words, what I want to say, or what I want to share with you, that generally when we study the TCA cycle, tricarboxylic acid cycle, which is the part of central energy yielding pathway. Okay, actually it starts with the glucose. Through the glycolysis, pyruvic acid is formed. From pyruvic acid, acetyl-CoA is formed. Acetyl-CoA travels through electron transport chain. These two carbon of the acetyl-CoA, they are channeled through this TCA cycle, and from this there are two steps where these two carbon atoms, they are removed or evolved in the form of carbon dioxide, and remaining carbons, they are recycled. Okay, but along with this nature, that is the catabolic nature, anabolic nature is there of this TCA cycle. these few intermediates, whatever I explained, those intermediates, they are involved in the synthesis process. Okay, that synthesis process is called as the anabolic process. That's why when any pathway, it functions like the catabolic pathway plus anabolic pathway. When it is a part of TCA cycle, generally routinely it acts as a catabolic pathway, okay, where the electrons are removed. From those electrons through electron transport chain, energy is generated in the form of ATP. That is the catabolic nature. But when such intermediates, important intermediate or important compounds, they are synthesized from the intermediates of TCA cycle, that is another nature, that is an anabolic nature. So when any pathway performs this dual role, okay, then that is called as amphibolic pathway. So I'll say TCA cycle is the best example of amphibolic pathway, because it not only is involved in the generation of ATP, okay, means energy generation, but it is also involved in the synthesis of certain biomolecules or compounds. Those are necessary for the cell. Okay, thank you very much.